So
Good, thank
you
you
so much for your questions.
Thank you so much, Sergey. And thank you, Diana, for such a wonderful talk. And we will sort of, move ahead. Next, we have Dr. Ryan Petrie. He received his PhD in cell biology from McGill University in 2008. Following a research fellowship in National Institutes of Health with Dr. Ken Yamada, he opened his lab in Drexel in 2015. At NIH, he used a combination of live cell imaging and biophysical measurements in single cells to discover a new pressure-based mechanism for cell movement. And he's also called Dr. Lobopodia because of that, I guess. His lab continues to refine this nuclear piston model, which we are going to hear today about. So thank you, Ryan. I'm sorry, Diana, we had to rush you. And there are a bunch of questions also in the chat if you would like to address it. And Ryan, the floor is yours.
Well, yeah, thank you so much for this great invitation. I love this this series, and I'm really thrilled to be here. Full disclosure, I called myself Dr. Lobopodia. There was no one else involved in that decision, but that was my own. So yeah, it's great to be able to give everyone sort of an update from Philadelphia on where we're at, kind of understand this nuclear piston mode of 3D cell migration. Of course, not all of you are probably familiar with it. So when I was in Ken's lab as a postdoc, I got really interested in understanding how the physical environment dictates how cells move. And our cell model of choice was primary human durable fibroblasts that we see here, migrating through our 3D in vitro model of choice cell-derived matrix, which is produced by the fibroblasts themselves when you grow them at a high density, and you can remove them, lyse them, and they leave behind this beautiful matrix material that the cells can migrate through, which we call 3D migration, but they also migrate on top. You can see sort of 2D and 3D migration in the exact same dish within the same experiment. So when I looked closely in these dishes, we saw this really interesting migration phenotype shown here at the top, labeled as a 3D cell, looking not very much like the cell that's migrating on the 2D surface just below it. This cell forms a uniaxial protrusion, and we discovered that it doesn't use lamellipodia at those tips of those protrusions, rather what we called lobopodia, which turned out to be a pressure-based protrusion. We further discovered that the reason why these cells are completely changing how they move is because in this particular matrix, they have a really big problem moving their nucleus. And it activates this actomyosin contractility that helps to pull the nucleus forward. And as it pulls the nucleus forward, it compresses the cytoplasm, generating pressure, and that pressure can then be channeled to form protrusions. So we call this the nuclear piston mode of cell migration, sort of summarized here. The reason why we called it a piston was because in a single cell, you could have the nucleus dividing a high pressure zone in front at about 2,500 pascals from a lower pressure zone behind at about 1,000 pascals. So the pressure was generating, the nucleus was generating the pressure, but it was also acting like a bit of a piston, forming a diffusion barrier between the front and the back of the cell, leading to this compartmentalized pressure, which ends up being the signature of the nuclear piston mechanism. Now, just to place this in context, because obviously we're all really interested in cell migration, there's a whole bunch of different ways for cells to migrate in 3D, I think widely recognized now. And back in 2016, just sort of based on qualitative information within the papers, we sort of segregated all these different modes based on contractility and adhesion. And we've got the friction-driven adhesions down here, the stable blebs, the osmotic engine, all at lower contractility, lower adhesion regimes. But when you get up into the high pressure, high adhesion modes of migration, this is where we find lobopodia-based motility. So this is what I am continuing to study here in Philadelphia. And the whole bunch of information here, just to sort of summarize what we know about nuclear piston-based motility, is specifically activated in non-linearly elastic materials. And this is, in linearly elastic materials, such as the cell-derived matrix we were just looking at, and that's a similar property to mammalian dermis, where we also see it happening. We know now a variety of primary human cells are capable of doing it, including dermal fibroblasts, cardiac fibroblasts, myofibroblasts, chondrocytes. In addition, we have the human breast cancer cell line MDA MB231 is also able to do it. Vivek Shenoy, Ovi Chaturi recently published that mesenchymal stem cells can use a form of the nuclear piston to help pathfind through certain types of matrix environments. Then looking at the mechanism itself, it absolutely does require adhesion. If you block adhesion and the cells are using the nuclear piston, the cells collapse, as we're seeing here in this movie, almost in sort of a singularity just in front of the nucleus. And it also similarly requires actomyosin contractility. If you inhibit myosin 2, pressure rapidly drops. And we also know that the cells switch back to lamellipodia-based migration. So it's a very plastic sort of mechanism. And when I first opened the lab in Drexel, we were very interested in understanding, well, is this pressure generating machinery the same or different from that generates traction forces? And through a variety of approaches that you can read about in the paper up here, we discovered that in fact, distinct tropomyosins define the traction generating machinery versus the pressure generating machinery. And that in fact, these things can be governed by distinct myosin 2 machineries. The pressure generating machinery seems to rely on tropomyosin 1.6 most of all, and that's enriched along with myosin 2 in front of the nucleus of cells that are using the nuclear piston mechanism. So that's kind of a snapshot of where we're at coming into this talk. And I just want to give you an update on answering these two research questions that I posed to my first two PhD students, Pragati Chengappa and Tia Jones, and the ones I want to share with you today. So the first is, and I should back up, we also know that vimentin intermediate filaments are very important in helping to pull the nucleus forward and cooperating with the actomyosin networks. So the first question is, well, how are vimentin and the actomyosin networks connected to transmit the pulling force of that machinery to the nucleus to generate that compartmentalized pressure? And that was Pragati's project. And the second question is, how does the cell activate this nuclear piston machinery to pull the nucleus forward in cells migrating through 3D matrices? And that was taken on by Tia Jones. So Pragati and I formed the initial hypothesis that the cytoskeleton cross-linker plectin is going to be the one that connects actomyosin contractility to vimentin to help pull the nucleus forward. Here we see vimentin right here. You can see how intimately it is wrapped around the nucleus. And in fact, it's known that plectin can help connect vimentin to the nucleus, to adhesions, and also to actin networks within the cytoplasm. So for us, it was just a question of, okay, let's see how this participates in the nuclear piston mechanism. So the first thing Pragati did was to knock down plectin and then check if that had any effect on the association of actin and vimentin in our human fibroblasts. And this is all on a two-dimensional surface. So the knockdown shown here on the left, and she used proximity ligation assays to get a measurement of how interactive the actin and vimentin networks were in control versus knockdown cells. And by eye, you can perhaps see that there is about a loss of a third of the connectivity in our partially plectin knockdown cells, quantified here. So we were able to sort of confirm that, yeah, plectin expression is required to help cross-link actin and vimentin filaments in fibroblasts on 2D. She then checked, so this is all happening on 2D, I just want to emphasize that. She then checked, well, what happens if you inhibit myosin 2 activity to these connections when fibroblasts are on 2D? And here she saw an even more dramatic loss of these connections within the cytoplasm. We didn't see much changes at the sort of light microscopy level. You really needed to do PLA to pick up the fact that these things were interacting and that these are dramatically diminished when myosin 2 is inhibited. She then checked if integrin engagement was going to be important in stimulating the formation of these connections between plectin and vimentin. To do that, she compared cells just plated on 2D glass in the presence of serum, so lots of fibronectin around, versus cells plated on PLL, where they have a harder time adhering. So here's just showing that, in fact, the focal adhesions that we all know and love can form very well on 2D glass, but on PLL, not so much. Here, detecting plectin-vimentin interactions via PLA, we get a much stronger signal compared to plectin-actin, interestingly, but that signal, that engagement seems to be lost if the cells are plated on PLL, blocking the integrin-mediated interaction, so measured here. And so this requirement for, it's happening on 2D, it requires myosin 2, it requires adhesions. We wondered if maybe the rigidity sensing machinery is playing a role in stimulating the formation of these plectin-vimentin interactions in our cells. So she plated cells on soft versus a stiff substrate and measured the amount of plectin-vimentin interactions by PLA and found that on the soft material, you actually got the most plectin-vimentin whereas on the stiffer material, there was a decrease. So it was surprising to us. I was predicting to be on the stiffer materials you get the most interactions, but we concluded that in fact, plectin-vimentin interactions can be governed by 2D substrate rigidity, but appear to be formed most in sort of this intermediate stiffness. Sorry, the softer stiffness compared to the stiffer materials. So all that being said, we know that they form a connection. Plectin helps form the connection between actin and vimentin. Is this at all important in the migration of our cells through these 3D materials? So Pragati took the plectin knockdown cells, put them in the cell-derived matrix, compared their motility, found that control cells move very nicely through the cell-derived matrix, whereas the plectin siRNA cells, they moved at about half the velocity of control cells. And when we looked at them, it appeared they're just kind of getting stuck within this material. And then further, we then checked the pressure, and we saw in control cells this compartmentalized pressure that's indicative of a nuclear piston mechanism that is then lost. You get this lower equalized pressure in the presence of the plectin knockdown, suggesting that in fact the plectin is required for the generation of compartmentalized pressure by the nuclear piston mechanism. So just to quickly summarize this first part, we kind of think of it as two steps. So plectin does connect these different networks, but the assembly of the networks might be controlled by something akin to the rigidity sensing machinery that we know controls traction force and adhesion formation in cells. It also seems to be being important in the formation of these actin-vimentin connections via plectin. But interestingly, it seems to respond most to soft substrates. And this is a softness that we'd find routinely in sort of our 3D collagen and cell-derived matrix gels. However, once the machinery is assembled, there's probably a second step that requires allows the pulling machinery to become activated to then generate that compartmentalized pressure to help move the nucleus through those linearly elastic 3D materials. So what is causing it to become activated to get the pulling forces to move the nucleus forward? And that was what Tia wanted to ask. So for the hypothesis she started with, we had a huge hint in a paper that we were really glad to be a part of from Alexis Lomakin, Science 2020, where they found that mechanical stress on the nucleus during 3D migration can actually increase stretch on the nuclear envelope, leading to signals in this diagram shown here on the right that increase contractility at the back of the cell at the cortex to help speed cells through the 3D matrix. I just want to note that this data that we shared as part of our effort in the paper was all from cells not using the nuclear piston mode of migration. They're moving in 3D, but these were fibrosarcoma cells that only use the piston when MMP activity is inhibited. And then, so we were wondering, okay, is mechanical stress on the nucleus, perhaps in the form of increasing stress in the nuclear envelope, also having a role in activating this pulling machinery by that pulls the nucleus forward to generate that pressure? And of course, work from Mathieu Peel and Lammerding and others has shown that the nucleus, in fact, can rupture when moving through confined environments. Just another obvious signal that the nucleus is under tremendous mechanical stress, and in fact, that the nucleus could become pressurized when moving through 3D, hence leading to that rupture. So we wanted to test the hypothesis, well, could mechanical stress on the nucleus be the deciding factor in activating this pulling machinery to generate that compartmentalized pressure? And the first thing Tia looked at was, well, is the nucleus more deformed when the piston is activated versus not? She compared cells migrating on 2D glass using lamellipodia, cells migrating on the surface of the 2D CDM using lamellipodia, and cells migrating in 3D CDM using the lamellipodia. And these are all HFF cells, the primary human fibroblasts. And she saw this clear trend that as we became more confined, and in fact, when cells are using the nuclear piston, that is when the nucleus is the most elongated, which is a measurement of sort of nuclear stress. However, when she took fibrosarcoma cells that you can induce piston activation by inhibiting MMPs, and that data is shown here where we have equalized pressure front and back in control cells. So they're in 3D, but they're not using the piston under these conditions. When we use GM6001 to inhibit MMP activity, we induce activation of the piston, shown here through this generation of compartmentalized pressure. But we saw no significant change in how deformed the nucleus was between those two conditions. So suggesting that deformation of the nucleus is not sufficient at all to activate the piston machinery. So what about nuclear pressurization? The fact that nuclei can rupture when they're undergoing 3D migration. Perhaps when the nucleus becomes highly pressurized, that could be the trigger for nuclear piston. Well, Tia adapted our pressure measuring protocol, which I can tell you a little bit more about at the end of the talk. And she compared a measurement in the cytoplasm versus the nucleus in primary fibroblasts on 2D glass. And in control cells, she found that compared to the cytoplasm, the nucleus is already compartmentalized, already pressurized relative to the cytoplasm in cells on 2D. And that partially requires myosin 2 activity. If you inhibit myosin 2, we don't see any drop in cytoplasmic pressure, but the nuclear pressure does drop. And she did some controls to confirm that she was measuring pressure inside the nucleus by selectively permeabilizing the plasma membrane versus the nuclear envelope, and showing that pressure was maintained when just the cytoplasma membrane was was permeabilized. And then really excitingly, when she inhibited MMP activity, that drove the pressure up inside the nucleus. So seeing that this change, when we activate the piston, we get this increase in nuclear pressure. Now wondering, okay, is that sufficient to activate the piston mechanism? And I'll just cut to the chase, and the answer is no. We, by accident, discovered that lipofectamine 3000 by itself, that's the cationic lipid shown here, when you add it to cells in 3D, it increases the pressure inside the nucleus. And when we do that intervention, there is absolutely no activation of the piston mechanism. We do not see the generation of the compartmentalized pressure under these conditions. So very regretfully, we had to conclude that mechanical stress in the nucleus in the form of deformation or pressurization is not sufficient to explain activation of the piston mechanism, kind of summarized here. And so what the heck do we do? And it took us a little while to kind of regroup. We designed a phosphoproteomic screen where we could just ask, what is the difference in the phosphopeptides when the piston's on versus off in our fibrosarcoma cell model? And Tia, we got a robust number of hits back that I am still having a great time sort of looking through and coming up with new ideas for what could be going on. But at some point, you got to stop dreaming and start doing. And so Tia looked through those list of peptides and sort of began to get a picture of where the RAS MAP kinase pathways seem to be a hotspot of changes in phosphorylation. And in gray are the proteins that came up as being changed in terms of how their phosphorylation changes when the piston is on versus off. And at the center here, we see RAS, RAF, MEK, and ERK, a pathway that's become increasingly understood to be very important in controlling things like collective cell migration, as well as movement of the nucleus. From the Gunderson lab, they have data showing that if you inhibit this pathway, you get the movement of the nucleus towards the back of the cell. So based on all this, we kind of had a fresh hypothesis. We wanted to check, well, does RAS signaling have any role in activation of the piston mechanism in our cells? And what we found was a little bit surprising. So here is Tia's data. She has the HT1080 cell. So this is migrating in that 3D cell-derived matrix. And under these conditions, they don't use the piston normally. So here the pressure is low and relatively equal. But when she inhibits GM6001 in these cells, she gets activation of the piston mechanism shown here, compartmentalized pressure. And then interestingly, when she just transfected dominant negative RAS into these HT1080 cells without inhibiting MMP activity at all, that was sufficient to generate the compartmentalized pressure and seemingly activate the nuclear piston mechanism. And then we did the inverse in primary human fibroblasts that use the piston normally in that exact same physical environment. Here, if we overexpressed either wild type RAS or constitutionally active RAS, both were sufficient to prevent piston activation in these cells. So the inverse. So we conclude from these two experiments that RAS signaling appeared to actually prevent piston activation in both fibrosarcoma cells and primary human fibroblasts. Very exciting. She also checked if the mode of migration was being affected in control cells staining with cortactin, which is our sort of molecular highlighter that lights up lamellipodial-based protrusion that we see here in the bottom. But in our wild type cells that are using the nuclear piston, they don't use lamellipodia as much. We don't detect it there, measured on this graph here. But when we express the wild type RAS or the tissue active RAS, both tended to increase the amount of lamellipodia forming. So pressure was going down and the cells were reverting to lamellipodia-based migration when the RAS signaling was inhibited. And then we follow that up by checking, well, what about ERK inhibition itself? And so here we used a chemical inhibitor with our HT1080 cells, and just like expressing dominant negative RAS, inhibiting ERK in these cells was sufficient to activate the piston. And that activation absolutely required myosin 2 activity. If we combined ERK inhibition with myosin 2 inhibition, we also similarly saw that the cells are no longer able to use the piston. And that these two movies are just showing you a consequence of when you inhibit ERK, you get these very blebby cells that is completely consistent with a nuclear piston lobopodia mode of migration. And then we also checked carefully that under these conditions where we're seeing an increase in myosin light chain at the back cortex. Again, just really wondering, is this a different mechanism than what was published by Lomakin in the science paper? And it appears that it could be. So we're getting this compartmentalized pressure, but we're not seeing that same increased signal at the back of the cell that could be indicating that it's contractility at the back that's helping to push the nucleus forward. We really think this is something that's specific to the pulling of the nucleus that could be acting in concert with the mechanism described by Alexis. So just to conclude this part, the mechanical stress in the nucleus doesn't seem to be sufficient for the piston mechanism, but when the cells are needing to activate the piston, we're likely going to see a decrease in RAS-ERK signaling that could be somehow integrated with MMP activity. We're still kind of working on that. And as that signaling goes down, that increases myosin 2 contractility in the pulling machinery in front of the nucleus, leading to the generation of compartmentalized pressure and the formation of lobopodia. And just one thing that's kind of really interesting about this result is, given how surprising it was to see the lobopodia type migration in the first place, maybe the fact that RAS transformed cells don't do it could be a hint that, because a lot of us use the transformed cells in these sort of cell motility studies, that just might have made it a little bit harder to detect in the first place. So we're excited to sort of follow this up and see what is controlling the RAS pathway and if it could explain these cell type dependent differences that we do see in a lot of cases. And these just movies are showing you how dramatically different you can get the cells to look when they're migrating in 3D, expressing just wild type RAS and reverting to this lamellipodia-based migration versus this high pressure lobopodia-based migration in our normal wild type cells. Oops, so with that, I'd like to thank you all for your time. I'd like to thank the lab, particularly Tia and Pragathi, who just recently graduated. Pragathi is now actually in the Gunderson lab as a postdoc, continuing to look at how cytoskeleton nuclear connections promote cell dynamics. And then we have the next generation of Matt, Jacob, and Bree carrying on the work. I'd like to thank Dr. Gerhard Weisch for his help with the plectin studies. And with that, be happy to answer any questions.
Fantastic. Thank you so much, Ryan. I also just want to start out by saying I really appreciate all the credit that you gave to your students throughout your talk. That's really, really nice to see. So thanks for doing that. So
No
a question
problem. That
from
was
earlier.
Ken Yamada taught me to do that.
Yeah,
that's something I learned from Ken.
A question from early in the talk. So I think, but it's still an open question from Ricard Alert, asking if pressure is higher in front of the nucleus, how is it pushing the nucleus forward? And, he would expect nucleus to be pushed from high to low pressure. So sort of the basic physical picture here, if you can give us some insight on that.
Well, yeah, absolutely. No problem. So that has been a bit of a mystery, is that this is a very unsustainable phenomenon, not just because of like, okay, the nucleus is actually fighting the pressure to move forward, but as the nucleus moves forward, why wouldn't water just get pushed out of the cytoplasm and then pressure drop and then the whole system kind of shut down? So when you inhibit cells with blebbistatin, the cell absolutely is pushed backwards and the pressure equalizes. So without that pulling force, the cell reverts to that sort of lower sort of energy level situation where you have non-compartmentalized pressure, and the nucleus is more at the back of the cell. But that doesn't answer the question: how do the cells sustain this over the long term? We've done a little bit of work with Vivek Shenoy now, who's gotten very interested in modeling this nuclear piston phenomenon. And what we're kind of circling is the idea that this, in concert with the pulling machinery on the nucleus to generate the pressure, water is being transported into the front part of the cell to maintain that high pressure while the cell moves. And then just quick idle speculation about why would you ever want high pressure in front of the nucleus, and besides forming a high pressure protrusion, one thing I'm thinking about is that maybe it helps prevent nuclear rupture. So if the nucleus tends to rupture from becoming pressurized, having a high pressure cushion in front of it could minimize that perturbation.
Okay, great. Thanks so much. We have a sort of a methodological question from Brunghung Yaha, asking, how did you measure pressure, for example, the 2.5 kilopascals in the front and the 1.5 kilopascals in the rear?
Oh yeah, sure. Let me just quickly share my slide here. So hopefully you can see that this is the micro pressure machine. We have it in the lab. You can buy it from World Precision Instruments. Basically, it's been described to me as a patch clamp, but for pressure. So you insert a needle carrying a current into the thing you want to measure the pressure of. The machine detects pressure-induced changes to the resistance of the circuit, and you get a very rapid, almost instantaneous readout of the pressure inside the compartment you're measuring. So we can stick that needle in front of the nucleus versus behind the nucleus and rapidly get these pressure measurements.
All right, great. Sophie, why don't you go ahead and unmute yourself and ask your question?
Hi, Ryan. Very nice talk. I have a question about extracellular matrix. In one of the movies, you show the cell moving through a purplish looking matrix. So you imagine the matrix as well, but I didn't see in that particular movie much changes to the matrix. So do cells, when they use this piston motion, do they modify ECM or do they just move in the available channels? Have you guys looked at that?
Yeah, I mean, it's been something that we've been thinking a lot about, because in collagen, where you do see a lot more matrix remodeling as the cells move through it, we don't see the piston mechanism. But in CDM, where we believe there's a lot more covalent cross-linking between the different fibers, which reduces their remodeling by the cells, we do see the piston. So that does seem to be a correlation. And we're still not sure exactly what's going on. But it does look like, because when we inhibit MMP activity in the fibrosarcoma cells in that matrix, that induces the piston, there must be some matrix remodeling going on that's helping the cells move through the material, that when you block it, it forces them to use the piston. So based on all that, I do think that the piston mechanism is maybe a way for the cells to move that is, when they are not remodeling the matrix so much, where they're chewing their holes through it or needing to completely rearrange the fibers. But when they can't do that, they seem to be able to use these other mechanisms that maybe more rely on protease activity. So it really is an open question, but those are kind of what we've been thinking about in terms of when we think about it that way.
Thanks.
Great, thanks so much. So are you okay, Ryan, to stay a few minutes longer? We have some more questions in the chat for you.
Yeah, I could stick around another few minutes, absolutely. Okay,
great. So we have a question from Trevor Ham, 2 questions about nuclear piston model. So the first one is about the cytoplasm. So you say that it's that you have compression of the cytoplasm, but he's asking, isn't the cytoplasm essentially incompressible?
Yeah, so you're right. I mean, fluid, of course, water is incompressible, but I guess what I'm thinking of there is sort of Guillaume Tiras's kind of idea where it's a sponge, a fluid-filled sponge. So when I say it's getting compressed, I guess I'm meaning the sponge containing the fluid is getting compressed, and that is then constrained by the plasma membrane, and that is what's raising the pressure.
Fantastic. And his follow-up questions related. So for a piston to work in the way that you're proposing, i.e. a hydraulic piston, doesn't there need to be a sort of a sealing ring between the piston, the nucleus, and the cylinder, the plasma membrane? Otherwise, that you would get this blow-by phenomenon where the fluid ends up being moved back and forth by the piston. So do you think that in your system, what is forming this piston between the nucleus and the plasma membrane?
Another fantastic question. So it turns out it's a very leaky seal. When we look at diffusion from front to back, it's slowed in cells using this mechanism. It's not blocked. So what the heck is going on? How could this even be possible? So this is where, again, Vivek Shenoy, some work he's been doing might be shedding some light on this. And what we're thinking is perhaps we're seeing a combination of adhesion, contractility dependent migration superimposed on the osmotic engine model. So the osmotic engine model, water is drawn in the front and pushed out the back. And if that's happening continuously in combination with this actomyosin machinery pulling the nucleus forward, we're thinking that could begin to explain how this, at first blush, untenable physical situation is actually maintained. But that's a work in progress, and that's Vivek Shenoy's, he's the one driving that effort. So that's really all I can offer at this point.
Awesome, fantastic. We have a comment from Ian Wong, just asking, so it seems like Vimentin's required for the nuclear piston mechanism, but noting that you didn't have any Vimentin negative cell types in the list on your introductory slide. Ian, if you want to weigh in further about your question there, or Ryan, any commentary to that?
Yeah, I can. No, there's not any Vimentin negative cells. We did knock down Vimentin, and we saw a disruption of the pressure draining machinery. We also knocked down Nesprin-3 and saw a specific loss of the pulling machinery. So those two, Nesprin-3 is known to interact predominantly with Vimentin intermediate filaments. So that's what we based this idea that Vimentin is required. We did begin to do a little bit of work of inducing EMT in epithelial cells to see if we could then induce a nuclear piston sort of competent phenotype, but have not successfully done that yet. We also haven't successfully actually shown Vimentin expressions increase, so that's a little bit of a puzzle at the moment. But there was a really interesting side result there I just want to mention, is that non-motile epithelial cells, before you induce EMT, are very high pressure. And when you induce EMT, the pressure drops and they become very motile and they form lamellipodia. So there's some consistencies between fibroblasts and epithelial cells in terms of how pressure combined with the ability to form lamellipodia. If you're interested in that, that was published in Physical Biology earlier this year.
Okay, fantastic. Anna Pasipara says that she has 3 questions, but I think only the first one is listed in the chat. So Anna, if you want to pop in with your next couple, but her first question is asking if you looked at expression of plectin by Western or PCR under your experimental conditions, soft and stiff. It looks like from the images, it looks like there's less protein.
Yeah, that's we've not really parsed that really finely. Really, all we do is just when we do the knockdown, we check, make sure the knockdown did its thing in terms of plectin. But we are following that up now because this paper is currently being revised for molecular biology of the cell. We're following that up now with a bit more careful look at plectin localization by IF. And I think if there are some differences, I think there's a good chance we'll pick it up as we do these revisions.
Yeah, because it looks from your images that there is like a different amount of plectin. And then since that protein also binds to intermediate filaments and actin, did you look at the microtubules if there is some differences in the distribution of microtubules in this shape and these cells that they're forming in the podia?
We have not seen any differences yet, Anna. But one thing was really interesting is that in the that data, that phosphoproteomics set that we got, there was several microtubule associated proteins. And we've never ruled out that microtubules absolutely have an important role in the piston machinery. But we're just not sure what they are yet. So we've got some now clues, I think, to go after. And I think we would sort of combine that with a much closer look at microtubule organization under these different conditions.
Okay. My third question is about the cell line, the HT180 that you used.
Yeah.
So you found in your proteomics that you have NRAS activation, but we know that those cells, they have by nature this mutation in that cell line. Did you try to do it in your fibroblasts, normal fibroblasts, just express your RAS and see if you have the same phenotype? Because the other one, you just inhibited something that was already there. But in the other side, in the other cell line, you don't have that, and then you still have the same phenotype. If you have upregulation of RAS, if, for example, your HHF1.
We did check that when we express a constitutively active form of RAS in the primary cells, it blocked piston activation. And so that was kind of the mirror of the story in HT1080 cells, where if you inhibit the RAS pathway, you can get piston activation. In the primary cells, if you activate, well, hyperactivate RAS, it completely loses the ability to use the piston. So that's as far as we've gotten on that so far. So it does seem to be happening in both. And where we're kind of thinking the connection could be is really through MMP activity. So that's something that the RAS pathway could be influencing the piston mechanism somehow through MMP production or secretion. But we haven't gotten very far on that next question yet.
Thank you. Very nice talk.
Thank you.
Good to see you.
Yeah, great to see you too.
Awesome. Ankita, did you see Nicola's question?
Yeah, I'll go ahead since I missed some of the questions. So Nicola's question is, does organization of cell-derived matrix change the probability of seeing piston migration?
It does actually, but it's not, we don't think it's necessarily organization. So the experiments we did even predate when we figured out how to measure pressure, is when we were just looking at protrusion identity. We found that if we trypsinize the cell-derived matrix, it maintains its organization. But when you add cells to it, they now can completely rearrange the fibers like to their whim. So that was kind of our idea that, okay, the cross-linking between the fibers is going to be important. And so we trypsinize it. When they go into that material, they no longer use the piston. They use lamellipodia, no problem. And if we cross-link the material after trypsinization with a chemical cross-linker, they then are forced to use the piston mechanism. So based on that, I've never thought it's necessarily remodeling per se, but more the cross-linking in the material that's going to dictate whether cells use Lipofectamine and nuclear piston or the low pressure lamellipodia phenotype.
Wonderful. I think that's the end of the questions. I really want to thank Diana and Ryan both for giving such wonderful talks. And there was like a flood of questions today, which is always amazing. And thank you everyone for such wonderful questions as well. I think we can stop the